So, separating signal from noise and metabarcoding data. So Ella described the general idea of DNA metabarcoding. And if you put this sort of in a larger context, you have this all of this molecular processing up here, and this sequencing machine will generate a lot of raw sequence data, as you saw from Ella's talk. We call those unique sequences that we get out of this workflow for amplicon sequence variants, ASVs. I'm going to use that. So I have a lot of ASVs from Sweden and from Madagascar, and then we still have a lot of noise in that data. So we need to try to reduce the noise level, and that's where we use the HAP pipeline that we developed ourselves. And of course, if you have developed methods, you know that the most important problem is coming up with cool acronyms. HAP, it stands for High Accuracy Pipeline for Processing Deep Metabarcoding Data. Deep stands for just a lot of metabarcoding data. So it's high performance also. And then HAP really consists of, we pieced existing tools together into our workflow, but we also added some components we've developed ourselves. And the major component we developed ourselves is this NEAT algorithm, and it stands for Noise Filtering Using Echoes, Evolutionary Signatures, and Abundance Thresholds. Well, good acronyms, I hope you agree. Okay, and the HAP pipeline design, it looks a little bit complicated, but it consists really of four major components. They're numbered here in the figure, and a lot of these things we can run in parallel, so we can process millions of reads easily, or ASVs. So these steps are chimera removal, taxonomic assignment, ASV clustering into OTUs, or these species units that Ella talked about, and noise filtering with a NEAT algorithm at the end. So I was going to just try to say something about each of these steps. So what chimeras? So maybe some of you don't know exactly what chimeras refer to in this context. So what we're trying to do, of course, before we do the sequencing, is we're copying the marker gene, the piece of DNA we're going to use for species identification. So you can imagine, and we're doing this in a sample. We have a mix of DNA sequences of different species. So imagine you have two different species here and their DNA sequences, and then we're trying to make a copy of the blue one here. And we just want a complete copy of the blue sequence. And then what can happen if two of these sequences are fairly similar to each other? They can sort of stick together, and the copying machinery can switch over to the other sequence in the middle. What, of course, at the end, instead of having a full copy of species 1 sequence, we get a hybrid, a chimera. So it's sort of an artificial cross between two species, and those two species can be completely distant from each other. It can be a fly and a springtail, for instance, a fly and a bumblebee. So with these look like undescribed species, we need to remove them. And we discovered that standard tools are supposed to remove these chimeras, but they actually aren't very effective. So we added an extra chimera removal step to really try to reduce this to a minimum. Then, so the next step in the HAP pipeline is to try to match each unique sequence or ASV that we have to a reference database and get a taxonomic name for that sequence. And we benchmarked 3 types of tools for this. K-mer-based tools, these 3, they're based on substring matching, so picking small pieces of the sequence and matching them to the database. Then we have alignment-based tools. They try to match the entire sequence against the reference database. And finally, phylogenetic placement tools. They actually have access to a tree where we know how the different reference sequences are related to each other. Unfortunately, we have a much smaller reference database here. So 50,000 sequences only in the largest tree we could find for this purpose. And we have about 100 times more sequences for the other methods. And then this is just to convince you that we did some serious benchmarking of these methods. And basically, we have a lot of different cases we're trying out here, and we know the ground truth, so we can see whether the methods are performing correctly or not. Here it's really simple case, and as you move to this corner, it gets more and more difficult. And beyond this black line here, it's impossible. So what we want to see is as much green as possible here, and it's getting more and more difficult as when you approach this boundary, right? When you got beyond the biome boundary, it's impossible, and the method should not try to give us a name. It should say that it doesn't know what it is. And if it still gives a name, it's red. So as much green as possible here, as much gray as possible here, and if you go through the details here, you see that syntax performs best. But we already see here that if we increase the size of the phylogenetic tree, these tree-based placement methods are going to be better, much better than these other tools. So, and perhaps I should mention, we just got funding, another Wallenberg project grant was recently announced to try to work with building these large insect trees needed for phylogenetic placement. Okay. So now we have some name for each of the unique sequences, but then we're grouping them into species, so related or very similar sequences that might represent the same species. We group them together. For this, we tested 3 different types of tools. They vary in their details, but they're based on some kind of similarity between the sequences, and also for DBOTU3 on co-occurrence, if they occur together in the samples. And we measure the performance by clustering sequences and testing against these species annotations that we had from syntax. Right? So in this case, in this upper benchmark, you want to be as close to this corner up here as possible to perform well. And the diamond is a really well-established algorithm wrestle performing on clean data and in contrast to noisy metabarcoding data. So this method didn't perform very well. These 2 methods actually both perform quite well. In this lower representation of the same benchmark, you want to be as close to this dashed line as possible. And again, you see that these 2 methods do quite well. And we opted for this method, Swarm, because it's really fast, robust performance across settings, and it's not very sensitive to noise if we have noise in the data. And finally, the NEAT algorithm. So the task of the NEAT algorithm is to remove sequencing errors, so small errors, just single bases that get where we make errors in the DNA sequencing. But most importantly, this problem with NUMS, so nuclear DNA of mitochondrial origin. So to try to explain that in simple terms, it's some organisms, including some insect groups, they make copies of this marker gene that we are using for species identification. And those copies, they start after some evolutionary time, they start looking like species that we have not seen. So we really need to remove them because they don't represent real species, right? And we have some off targets, so just random hits that don't represent the target marker at all. So we use a 5-step approach for this, so 5 different steps in the algorithm, and then we benchmark it against 3 different methods that have been proposed in the literature: LULU, cool acronym, isn't it? HMM, and abundance filtering. And here I'm showing 2 different types of benchmarks. The first benchmark, you see the HMM method in orange here, the abundance method in blue here, our method in green, and Lulu actually is doing quite well in this benchmark. You want to be as close to this corner as possible, right? Here is another, this is a very good benchmark because grasshoppers are just a nightmare. They have so many copies of this marker gene, and we just need to remove them because they are not real species, right? So we have encountered in our samples 20 species of grasshoppers, and the raw unfiltered data has 1,400 clusters of grasshoppers. So we need to remove that noise, right? HMM doesn't work very well here. Lulu is not very effective either. NEAT is quite effective actually in removing that type of noise. So we think it represents a big step forward in reducing noise in these data. And finally, I wanted to show you what's good is that we have, as has been mentioned already, right? We can compare with what we know about the Swedish insect fauna. And the Lepidoptera, that's one of the groups we know the most about in the Swedish fauna. So we don't expect to find a lot of new things. And that's also when we look at the data we have, we see that of all the Lepidoptera species, about 2,800, this part is what we retrieve in our malaise traps. These species, we haven't found those. And then we have just a few things here that either are new things or they represent noise. So really a small amount of noise. And then it turns out actually when we look at these things that potentially could be noise, actually some of them represent new finds. Well, maybe I should say before I move on to that, I'll give an example of that. If you look across all of the families of Lepidoptera, the pattern is about the same for all of these groups. So we find about half of the species in the IBA of the Swedish species in the IBA catch. It's true for the big butterflies and moths, Macrolepidoptera, and it's also true for the tiny ones, Microlepidoptera. And this is an example of something new that we found in Lepidoptera, which is supposedly very well known in Sweden. So if you live around the Stockholm area, you've seen oaks attacked by these things. These are small Lepidopterans. Larvae feed between the upper and lower surface of the oak leaves. And the oaks look horrible when they're attacked by this. Right? And we thought that there was this species that caused this. But it turns out actually this species is two different species. And one had not been described. It was described last year. We were the first, I think, to realize this. And we had the most extensive data on the distribution of that species. It's apparently an invasive species that's been introduced here and probably responsible for these outbreaks. Okay, thank you.